So as you have seen, the first step of initiation is the formation of closed complex. So the next step is the transition from closed complex to the open complex. So transition to the open complex involves structural changes in both the RNA polymerase and in the promoter DNA. The initial binding of RNA polymerase to the promoter DNA in closed complex leaves the DNA in double stranded form. You have seen that when closed group is formed, the DNA remains in the double stranded form, and polymerase binds like this. So the DNA is still in double stranded form in closed complex. The next stage in initiation requires the enzyme to become more intimately engaged with the promoter in the open complex. The transition from the closed to the open complex involves structural changes in the enzyme and the opening of the DNA double helix to reveal the template and non-template strand. So when the strand is DNA double helical strand is opened, two strands are formed. One strand is template strand and the other one is non-template strand. So these two strands are revealed during this opening up. Template strand is the strand which will be transcribed. This melting occurs between the positions -11 and +2 with respect to the transcription start site. So at the transcription start site, there are two positions on this DNA strand. One is recognized as -11 position site, and other is +2 site. In case of bacterial enzyme bearing sigma 70, this transition is often called isomerization. So you have seen the structure of RNA polymerase. There was one subunit which is sigma subunit. So bacterial RNA polymerase contain this sigma subunit, and this sigma subunit is called sigma 70. This transition doesn't require energy derived from ATP hydrolysis and is instead the result of the spontaneous conformational change in the DNA enzyme complex to more energetically favorable form. So, you might be knowing about the enzyme substrate complex, and during enzyme substrate complex, there is some transitional change in the structure of enzyme, which then brings about catalysis or brings about the reaction. So the energy required for this transition, it doesn't come from the ATP hydrolysis. Rather, it comes from this change in the conformation of enzyme. Two bases in the non-template strand of the -10 element flip out from their base stacking interactions. So in the double-stranded DNA, in the center of strand, the bases, they interact with each other. The bases of two strands, they interact with each other, and this we called base stacking interaction. So in the non-template DNA strand, these two bases, these flip out from the strand. They are inside the strand and they flip out from the strand, and after flipping out, they insert into the pockets within the sigma protein, where they make more favorable interactions. So this is double-stranded DNA. It has opened up after the formation of open complex, and then the two bases from the non-template strand, they flip out, and when they flip out, Here is RNA polymerase attached here. And in this RNA polymerase, there is one sigma factor. So these two bases, they are inserted into the sigma factor, and where they make some more favorable interaction. By stabilizing the single-stranded form of the -10 element, these interactions derive melting of the promoter region. So now the promoter region melts. The melting means the opening up of two DNA strands. Isomerization is essentially irreversible, and once complete, typically guarantees that transcription will subsequently initiate. So once these events happen, they guarantee that now the transcription will take place. Formation of closed complex, in contrast, is readily reversible. So the attachment of RNA polymerase to the double-stranded DNA and formation of this closed complex, it can be readily reversed. So, but the formation of open complex and then the happening of some events, they cannot be reversed. The examination of the structure of the holoenzyme in more details reveals the active site of the enzyme, and this active site is made up of regions from both the beta and sigma beta prime subunits, and it is found at the base of the pincers within the active center cleft.